developmental morphology. Another definition of morphogenesis, the study of various factors which affect the organic form and describe the growth pattern of the individuals, which means how the plants developed. During the tissue culture technique, if you select the explant may be a single cell or the explant may be a tissue, the explant may be a protoplast, the explant may be a cytoplasm, whatever it may be, it regenerate and differentiated into plant parts and finally developed as a complete plant. This total process called as morphogenesis. Whatever it may be. Hello? PRAOA host. Right, thank you. Right. So in vitro morphogenesis involved anatomical and physiological events. The both are involved in growth and development of an organism. Growth and development of an organism, which included in the morphogenesis studies. Let's see the factors. I think definition clear. Let the morphogenesis simple, the formation of complete individuals or complete vegetative body from the explant. Already told you the explant may be a single cell or tissue or protoplast, cytoplasm or any part of the plant. So that is the brief definition about the morphogenesis. Now, what are the factors involved in morphogenesis? The same factor which are involved in tissue culture technique here involved. Maybe the all macro and micronutrients required. All macro and micronutrients required. And hormones, mainly cytokinin auxins. These are the morphogenetic hormones. Cytokinin and auxin ratio involved for differentiation of the plant parts or differentiation of the tissue to develop a individual plant. Apart from that, various concentration of T4D auxins and physical factors also required, such as light, temperature, proper pH. The chemical factors like carbon, nitrogen ratio, oxygen content, and sugar concentrations, optimum concentration also required. Already told you the growth hormones like auxin, cytokinin, gibberellins, and abscisic acid. So these are the requirement for the morphogenesis, right? Some of the morphogenesis terminology. Let's see some terminology. Yeah. Right. So during morphogenesis, some of the portions are developed. For example, the property of totipotency. First of all, totipotency already told you yesterday. The totipotency property means the plant cell have the ability to regenerate into plantlets on plant tissue culture medium. Already we discussed yesterday how the culture media prepared. How the culture medium prepared. And what are the techniques involved in it also we discussed yesterday. Right? So that is totipotency. The toti simple definition, the hability. Actually, we know the explant developed into complete plantlet. In this process, what are involved? What property involved? There is one important property that only present in plant, that is totipotency, which means ability. The plant cell can ability of regenerate. That capacity called as totipotency. Based on totipotency, cytodifferentiation takes place. The second one, just one. Cytodifferentiation takes place. Cytodifferentiation means cellular differentiation to morphogenetic development of higher plants. That is called cytodifferentiation. That means cellular differentiation. Second morphogenesis part is xylogenesis. Xylogenesis, differentiation of parenchyma into localized secondary wall thickening in xylem tissue system. That means the parenchyma cells, if you take parenchyma cells as explant, from the parenchyma cells, xylem tissues are developed. If the xylem tissue part developed, this part of the morphogenesis called as xylogenesis. Genesis means production or formation. Xylem cells formed from parenchyma. So here the xylem formed, that's why called as xylogenesis. The cells developed called as cytodifferentiation or cytogenesis. Next, rhizogenesis. Rhizoids means here roots. Rhizogenesis. Rhizoid means roots. The development of root from the explant or callus. Explant or callus. Development of roots called as rhizogenesis. Next, callogenesis. Callo, shoot, regeneration of shoot from the explant. That is called callogenesis. If the roots are regenerated, that is called rhizogenesis. Shoots are developed, that is called callogenesis. Next, callus. Callus means unorganized mass of cells. Unorganized mass of cells that originate from an explant. Already told you about the explant. In vitro culture, explant lost their histological status. This is known as de-differentiation. Callus obtained from the protoplast is called protocallus. Whatever it may be, here the callus may be developed from the any type of explant. The one of the example, if the callus developed from the protoplast, that is called protocallus. Organogenesis. That means instead of cellular differentiation, instead of xylem elements formation, instead of root, instead of shoot, instead of callus, the in vitro, the regeneration of shoot, root, floral buds, all. These are called organ. That's why it's called as organogenesis, which are developed from the explant. Anyone here, any morphological structure developed from the explant only. Or explant, here the explant direct organogenesis. Direct organogenesis means here the organs directly involved. Or from the callus. And yesterday discussed another one is somatic embryogenesis. Somatic embryogenesis. What is somatic embryo? The embryo formation instead of from the reproductive cells. Actually, in general, the embryo formation after fertilization, a fusion of male and female gamete. In this process, first zygote formed. The zygote further developed into embryo. But rather than that process, here the somatic cells, the somatic cells selected from the embryonic cell. Here the embryonic cells, the plant embryonic cell act as a explant after culturing. This is developed into embryo. This is called somatic embryo. So these are the some morphogenesis terminology. And another one is androgenesis and gynogenesis. Androgenesis means andro means androecium. What is androecium? Stamens. In vitro differentiation of colors or plantlet from the microspores, where the microspores are developed already in the mean itself given androgenesis. Andro means already told you androecium. What is androecium? Androecium means stamens group. What are present in what are the parts of the stamen? Stamens have filament and anther. Filament and anther. Inside the anther, pollen grains are present. In androgenesis technique, you may select from the whole anther or you may select from the microspores or pollen grain as a explant. If you select microspore or anther or and sorry, microspores or pollen grains and anthers, whatever it may be, if you select it as an explant, this explant after tissue culture technique, after inoculated into the medium, it is developed into a complete plant. This is called androgenesis. Next, gynogenesis, gynoecium. In vitro regeneration of haploid plantlets from the unpollinated female gametophyte or embryo sac cells by culturing ovules is known as gynogenesis. So gynoecium parts already, what are the gynoecium ovary style stigma? In this is explant utilized from the ovary, maybe our ovule, or inside the ovules, embryo sac develop, or the explant may be ovule or embryo sac. If the morphogenesis developed from the ovule or embryo sac, that is called gynogenesis. In 1976, Sanyo first reported haploid plantlet regeneration via gynogenesis in barley. Gynogenesis has become another technique for haploid production in plant tissue culture. Here the haploid production not only for gynogenesis and also from androgenesis also we can get haploid plantlets. So this is the in vitro morphogenesis types of terminology. Next, types of culture. Morphogenesis. Right. Types of culture. Different type of cultures are there. Mainly based on the explant. Here, cell culture, organ culture, callus culture, or protoplast culture. And another one, cytoplasm culture also there. Pollen grain culture also there. So these are the different type of culture. First, let's discuss about the cell culture. The cell culture is the culture of isolated individual cells, which is obtained from the explant tissue or callus. In cell culture, we utilized most probably one single cell. Right? Let's discuss cell culture. The cell culture to culturing of the cells required the suspense culture media. Suspension culture media instead of agar media. Agar somewhat solid medium, but instead of agar medium in cell culturing utilized suspension culture medium. Suspension culture medium. This suspension culture medium is simply multiplying the single cell at higher rate in liquid medium. So agar medium somewhat solid nature, but in suspension culture in liquid medium, we can take the all nutrition in the form of liquid, right? So the liquid medium continuously agitated in an orbital shakers. This liquid medium introduced into the orbital shakers. It's continuously, maybe 2 to 3 days. So this is the popular culture method for many industry to extract certain component of the plant cells. So do you want to get number of plant cells? The first you prepare the liquid nutrient medium, that is called suspension culture. After suspension culture, to the suspension culture you add the collars. You inoculated collars and place it inside the orbital shakers. What happens if you introduce into the orbital shakers? It keeps shakes continuously. If you keep shake continuously, the exact pressures form on the tissues or the collars. So agitation of the medium also exert. That means due to the shaking, due to the shaking of the culture, and the exact mild pressure falls on the collars or tissue. As a result, breaks into smaller cells and single cells. This is the this is takes into the flask. For example, this is the flask. So flask already these are the nutrients, liquid nutrients. In the liquid nutrients, to the liquid nutrients, you introduced or inoculated the collars. Already told you the mass group of cells collar. It made up a number of cells. And this conical flask placed in orbital shaker for 2 to 3 days. Due to the shaking pressure, this collars separated into cells, separated into number of cells like this, separated individual cells. So already tell you in first line, the simple multiplying single cell at higher rate, single cells. So from the collars, single cells developed from the higher rate. So it's break into smaller cell pieces and single cell. This hesitations to maintain the uniform distribution and movement of the cells in the medium. So later, due to the shaking, these all cells spread entire the medium. So this is the important procedure for the cell suspension culture. I hope you understand why the provide constant agitation to medium. Why we provide constant agitation to medium? Because of create a pressure to breaks into smaller pieces or single cells from the callus or tissues. Then how do we get the single cell? First of all, the single cell we get from the callus. Single cell we get from the callus. Already yesterday we discussed how the callus formed. During tissue culture techniques, the mass group of cells are formed, that is called callus. After developed callus, this callus transferred into the from liquid medium in clinical flask and placed in orbital shakers. Now this callus divide into single cells, group of individual single cells. Now callus is already told you, the callus is an irregular mass of plant tissue that do not have any structure. There is no root, no shoot, no leaf, just mass group of cell. Even though there is no clear structure, but it has the capability to differentiate it into different plant organs. This is carried out by the one important property that is already told you, totipotency property. So we can obtain different colony from the single explant. First, to produce the colors, we selected the explant. After the explant, you produce the colors. These colors transferred into the conical flask liquid medium, and you applied the shakers, orbital shakers. As a result, single cells separated. So this colony from the same explant might show the variations. Of course, explant same. Of course, explant same. From the same explant, different type of colony, various colony form. From the various colony, you separated different cells, different type of cells. But after separated into the cells, they may not uniform. There some variation maybe occurs. So these are the different cells formed. Of course, these are the different type of cells. But what is the mother? The mother is same, the same colors, same explant. But during the technique process, some variation takes place in terms of color, texture, and also chromosome number differentiation. Gene mutations also occurs in the cells. The cali may be compact and friable in texture, light, dark in color, and so on. Right. The one of the interesting facts about the callus culture is that you can multiply them for an unlimited period of time without any specific differentiation. You can do this by periodically by subculturing or Fresnerian continuation, adding auxins and cytokinins. So this is cell culturing. So mainly the cell culture process. First of all, you selected you obtained the cells. How can we get the cells? The cells can get from the callus after applied with shakers, orbital shakers. How can you get callus? Callus get from the explant. Here from the explant callus, from the callus cell separated. So this is callus single cell. And let's see the types of the callus culture. Here the two main types of suspension culture are there. One is batch culture, second one is continuous culture. Let's see about the batch culture. Right. Types of cell suspension culture. Already told you two type of, one is batch culture and continuous culture. First, let's discuss batch culture. Batch culture is a fixed volume of nutrient culture medium. It's fixed for any medium. In this culture, cell division and cell growth coupled with increase in biomass occurs until only for the formation of biomass. The main factor involved is environment. That is the limiting factor to production of biomass of cells. The cells exhibit the following 5 phases of growth when the cell number in suspension culture is plotted against y-axis and x-axis. If you take graph, one is x-axis, y-axis. So here During a batch culture, log phase, log phase, linear phase, stationary phase, declaration phase, and again final stationary phase. Right. The log actually the first phase in batch culture in first phase preparation of cell for divide. Here in first phase all metabolic there is no growth. Log phase means there is no growth. Let's change color. There is no growth. Just starting like this. Stable. What happened during the log phase? During log phase, all metabolic activities developed. These metabolic activities, these metabolic properties required for the next stage. That is log phase stage. So in this stage, in log phase stage, no cell division, no cell volume increase, no growth, only ready to division. The all requirements gathered, all requirements prepared during the log phase. Next enter into the log phase, log phase. So where the rate of cell multiplication is highest. Now here the cell divided and growth also takes place. Growth also takes place like this. To grow continuously. Because in first phase already all are prepared to growth to division. The growth and division takes place in next stage. But in first phase, instead of division, instead of growth, only the metabolic activities are developed. As it developed metabolic activities, the next stage raised up in highest manner. So the rate of cell multiplication is highest in log phase. In next linear phase, it is represented by slowness in cell division and increase in cell size expansion. In log phase, cell division and cell volume size also increasing. But in linear phase, somewhat down. Slower rate. The linear phase is represented slower rate in cell division, increase in cell size expansion. Next in constant stationary phase. The stationary phase was constant numbers of cell. Constant like this. No increase, no decrease. No increase and no decrease. Actually in linear phase somewhat down here. And next no increase and no decrease. But what happened in stationary phase? During stationary phase, some of the cells are destroyed and the same manner new cells are regenerated. That's why it is stationary. That's why no, for example, after log phase and linear phase, there are different type of cells are formed. For example, in this group of cells, for 100 cells are destroyed. 100 cells are destroyed, and immediately another 100 cells are formed. What about net content? The net content is 0. 100 destroyed, again 100 formed. That's why it is called stationary, stable, maintained. That's why stationary stage. The declaration, it is actually the declaration, it is characterized by the decrease in cell division and cell expansion. Decrease cell division, cell expansions, decrease here. Finally, death phase, stationary, after stationary, death phase. This is end. Death phase or finally stationary phase. This is happening in batch culture. Right? And in batch culture, so first let's discuss again. Log phase, stable, no growth, just metabolic activity takes place in this stage. A log phase, due to all the metabolic formation takes place, it is shows the rapid multiplication. So that's why it's a growth rapidly occurs. Linear slowdown, stationary, stable, and again slow down and finally death occur. So this is batch culture. And in batch culture, it is a closed system of culture whereby we can grow cells in fixed amount. Already told you the fixed amount of culture medium under appropriate conditions. For example, this kind of, let's take using flask, conical flask, the fixed amount just to 250 ml. You can treat this first flask when cells inoculation, further flasks in suspension. Okay. For each subsequent subculture, the small eloquent is taken from the initial suspense and then transferred to fresh medium. In batch culture, another important one is, for example, this is one conical flask. You prepare the medium. Next, you prepare another suspension. So first of all, in first conical flask, some of the cells are developed, some of the tissue, some of the colors developed. Later, these are transferred into the another new medium, subculturing. For each subsequent subculture, small liquid taken from the initial suspension, that is first suspension, and then transferred to a fresh medium. So this is happening every time. Here, the major drawback of the batch culture, cells grow up to a certain point, but not completely, just to development of biomass. After finally, the death occurs. And then cell growth becomes stationary. Finally, it's stationary, stable, or maybe death occurs also. In this stationary phase, number of cells and size of number of cells and size of cells remains constant. So what happened in batch culture? There is no further growth. Only in log phase shows the rapid growth occur. Later, it settled at stationary stage, stationary level. When cell reach stationary phase, either because exhaustion of some growth factors or accumulation of toxic metabolites. Why stationary phase occurs? Because of exhaustion of some growth factor or some toxic metabolites. Due to the toxic metabolites, there is no further growth. Exhaustion of there is no tightness of the regulators also, there is no further growth. So, however, you can avoid this phase by providing fresh media at faster rate. That means actually this is first media. When the first media, the group of cell reached the stationary stage, before reach the stationary stage, you can remove it. Medium transfer to fresh medium. Again, continuously log phase and log phase. When before reach to the next stationary stage, again transfer to another fresh medium. Again fresh medium. So this continuously transfer to the fresh medium to fresh medium. So this is the batch culture. And another method is, as I said just now, the continuous culture. Let's see the continuous culture. In this type of cell suspension, you can maintain constant phase of cell growth. There is no rapid growth. That means if you take in batch culture, first stable, rapid growth, stable, and again down. So this is all happened in batch culture. But in continuous in growth, the maintain constant phase. The development slowly developed the constant rate. Here the fresh medium is continuously added. and left over the nutrient and metabolites end product are constantly removed from the first medium. What happened here? Here in batch culture, conical plus. So this media or this growth of tissue callus again utilized. But in continuous culture, you utilize this. This is completely avoided. Then every time you use fresh medium and fresh callus. Every time fresh medium and fresh callus. Thus, in this method, you avoid the detoxification. Already told you, due to the detoxification, there is a formation of constant stage. The growth retarded. But due to this continuous process, the continuing removing the medium, you can avoid the detoxification and the overcome the drawback of the batch culture, right? And where it is, and this continuation cultures has again two types. Before chemo start and uh, turbo start, let's discuss about closed continuation culture and open continuation culture. So first, in this culture, there is a frequent addition of fresh medium and draining out of the used medium. This is very important. In this medium, frequent addition of fresh medium and draining out of used medium so that the culture volume is normally constant. These culture are carried out specially designed culture vessels. The separate culture vessels, it is two type: closed continuation culture and open culture. Closed continuation culture. In this culture, the cells are retained while the inflow of fresh medium is balanced with the outflow of corresponding spent medium. The cells present in the outflowing medium are separated and added back to the culture system. So in closed system, all the retained cells in first medium balanced with outflow corresponding spent medium. The cells present in the outflowing medium are separated. When you outflowing, that means now when you remove the medium, some of the cells separated and added back to the new culture medium system. As a result, there is a continuous increase in the biomass is closed continuation culture. And open continuation culture, in this culture, the inflow of fresh medium is balanced with the outflow of the volume of spent medium along with the cells. In addition of fresh medium and culture always are adjusted that the culture are maintained indefinitely at constant growth rate. This is open continuation medium. And another one is synchronization suspension culture. Synchronization of suspension culture. Synchronization. So what is synchronous? Synchronous, all are the same. That means if you take the group of cells, in group of cells, cell size, cell volume, cell growth, and cell division stages also at the same level. But what happened during the culturing process? In normal situation, in cultured plant cells very great in size, shape, and cell cycle stages also. And it's said to be the asynchronous. This is called as a asynchronous process. It is not a synchronous. That means, so this is the group of cells. Group of cell. One is small cell, one is large cell, and even it is similar size of cells also. In one cell G1 stage present. In one cell J stage shows. One is M stage, one is G2 stage. So the variations among the culturing group of cells. So this is called as a asynchronous, this type called as asynchronous. But due to the variation, asynchronous condition, these variations not suitable for genetic study, genetic and biochemical and physiological, any type of study, it is not useful. This is not useful because as it variation in cells, how can we get study? So it is impossible to study if we in asynchronous condition. So we should maintain the synchronization, the culture medium. To maintain synchronization, there are the important methods are involved. One is physical method, another one is chemical method. First, let's discuss about physical method. That means we make it balanced. The culture medium cells, we have to make it balanced. We have to make it synchronized. The synchronized by physical method. The culture growth influenced by physical parameters such as optimum light, temperature. When we properly use the light and temperature, mainly the properties of cell like size, suspension, culture subject to low temperature, maybe low temperature. If the cell any variation in cell size, we apply the low temperature. Due to the low temperature, all cells are maintained the same size, almost around the 4 degrees centigrade. This 4 degrees centigrade shocks to utilize for maintaining synchronizations. Now the cell suspension selected for the any type of study. Right? This is the physical method. So in physical method, to maintain synchronization, utilize proper light and temperature. Here the temperature may be around 4 degrees centigrade. Right? So this is one physical method. Let's see the chemical method. Chemical method. The chemical method for synchronization suspension culture include the use of chemicals. With help of these chemicals, the cell cycles can be arrested at a particular stage. So for example, one cell G1, another one S or G2, different type of stages are there. During different stages, if we apply some chemicals, it maintains. If we apply the chemicals, due to the chemicals, the stages are arrested at particular level. The chemical inhibition, the DNA synthesis, such as, for example, DNA synthesis inhibition during the cell division, inhibition of DNA synthesis, 5-amino uracil, hydroxyurea, and 5-fluorodeoxypurine were added to the culture, resulting in the accumulation of cell at G1 stage. When we apply 5-amino uracil, hydroxyurea, 5-fluorodeoxypurine, automatically the cell stages arrested at G1 stage. So in this group, all cells stable at G1 stage, and it is turns into synchronization. The colchicine is strong inhibition to arrest the growth of the cells at metaphase. In interface, maybe G1, G2, yes, you utilize some chemicals, 5-amino uracil, hydroxyurea, 5-fluorodeoxypurine. But in already the interface completed in the different type of cell present in division stage, prophase, metaphase, anaphase, telophase, then we use colchicine This calcium strong inhibitor to arrest the growth of the cells at metaphase level. It inhibits the spindle formation during the metaphase as a result of chromosome doubling. What happens if spindle formation takes place? If any cell spindle formation takes place, the cells enter into the metaphase because the spindle apparatus arrange the chromosomes at the middle of the cell. As a result, enter the metaphase. After that, this spindle fiber contract each other, shows contraction nature each other. As a result, due to draw opposite direction, enter the anaphase level. After disappear this apparatus and enter into the telophase. So they all occur if the spindle fiber formation. What if if the spindle fiber absent? These further stages all are retarded. The further stages all are absent. So the activity of do not formation spindle fibers done by colchicine. The colchicine must be done for short period of during expansion phase. Another chemical method is starvation. Starvation means when an essential nutrients or growth promoting compounds are deprived in suspension culture. As a result, stationary growth phase. That means, for example, in a nutrient medium, in a liquid nutrient medium, there are several nutrients are present. Where this nutrient are utilized by the colors and growth takes place. So if we remove the nutrient, some of the nutrient from the medium, what happened? There is no nutrition to grow colors, to grow culture. Now it is entered the stationary. That means all cells are get reached the stability. That is stationary. Sorry, that is called as starvation. So this is the chemical method. Next one. And so synchronous physical method and chemical method also completed. Next. Now methods to assess the viability of cells in suspension culture. What are the methods? What are the methods involved in this? Let's see the methods. One, phase contrast microscopy method, utilized for assess the suspension culture, whether it is good or bad. And tetrazolium salts reduction, fluorescence diastate method, Evans blue staining. These are all utilized for the assess viability of cells suspension culture. That means after utilizing from culturing medium from the colors, after separated the cells, we need to check whether these cells have the viability or not. So the group of cells, to check the viability, the utilize some of the methods. First of all, phase contrast microscopy. Here the scientists evaluate the viability by observing healthy cells components and movement within the cell obtained from the cell suspension. Already cell suspensions, already the cell suspension means already the nutrient medium the spread over the cells. Let's check these cells, this group of cells, whether they have the viability or not. If we use contrast microscope, when we observing, we can, based on the cell components and movement of the cell, we can decide it, this is viability cells or non-viability cell. That is one method. So in this method, we can use phase contrast microscopy. And another method is tetrazolium salts reduction. With this method, scientists can measure the respiratory efficiency of the cell. When we use tetrazolium salt, the level of respiratory, we can measure the level of respiratory. So based on the efficiency of respiration, the respiration present, that is the viable cell. There is no proper respiration, that is decided as a non-viable cell. That is the tetrazolium salt reduction. And another method is fluorescence diestate method. With this method, scientists can measure the percentage of viability of the cells. It works on the principle of illumination of the cell fluorescence green color. If you apply the fluorescence light, some green color reflect. The illumination of the cells green color. If the cells green color illuminated, that considered as a viability cells. The rest of the all non-viable cell. So in this method, we can calculate the percentage in the group of cells. How many cells viable, how many cells non-viable, we can estimate in this method. Next, Evans blue staining method. In this method, scientists treat the cells with Evans blue chemical. This Evans blue chemicals also utilized for the viability pollen grain viable test also. The stain where the dead cells take up the stains and the viable cells remain unstained. That's it. After formation suspensions of group of cells, you separated the cells, suspension group of cell from nutrient medium and add Evans blue. When you apply the Evans blue chemical to those cells, in the group of cell, if any dead cell present, these dead cells taken up the stain, which is blue in color, somewhat violet blue color. If you observe the violet blue color cells, they are considered the dead cells non-viable and which are present unstained. As such, that cells considered as a viable cells. Right? The next one, Kalas culture. The next in it, Kalas, just wait. Next in it, Kalas culture. Already told you what is Kalas. Kalas is the undifferentiated and unorganized mass of plant cells. It is basically a tissue which usually forms on wounds of differentiated tissues or organs. Callus culture cells are parenchymatous in nature, although they are not truly homogeneous. That's why there's some changes, some variations also occurs in this. Callus formation in vivo is frequently observed as results of wounds at the cut's edge, stems or roots. If you cut the plant, we can observe after a few days the group of cells are formed. That is formed in vivo, not in vitro. The invasion of microorganisms or damage by insect usually occur those callus as it exposed out. There is a chance to infect with microbes. And apart from that, auxin to cytokinin ratio in plant tissue culture medium give rise to unorganized growing and dividing mass of culture. This is formed in vitro condition, and it is also formed in vitro and also in vivo. For example, in natural, generally, it's imbalanced apparent in the auxin cytokinins. We already told you auxin cytokinins to utilize for the morphogenesis. If it is utilized in proper way, but if it is applied improper way, it is unorganized growing into divided into mass cells, the group of mass cells. So the callus culture are often broadly classified being either compact or friable. So let's discuss later. So this group of cells grown on solid medium, the callus through solid medium from the plant or explant. So it may initiate from the explant like root, stem tips, leaf, flower, fruit, any multicellular plants. Already told you earlier, the callus development from the explant. From the explant, after formation callus, the callus may be transferred to solid medium for further development. If you want to get a cell, if you want to get different type of cells, instead of solid medium, as I said earlier, you transfer the callus into the liquid medium. That's it. And we will discuss later about in detail callus culture and protoplast culture, cytoplasm culture later. Thank you all.